Okay, so I'm glad that I can talk about our work now after having given 2 different talks on the area in which we work. So I put quite a bit on my plate today. We'll see how far we can go. But essentially, I'd like to talk about the important aspects of morphogenesis which touch upon the mechanical control and the biochemical regulation of morphogenesis. And I think this first slide, so I'm skipping some introduction because, we are at the end of the course and at least a lot of people have had some introduction to a lot of what I will talk about today, although some people will have less introduction, but although I talked about information in development, this is for the black hole physicists of the audience. Anyways, I think 2 important challenges in understanding development, and in particular morphogenesis, are stated in this slide. And that is what really catches me a lot into development in general, and morphogenesis in particular, is the fact that we would like in the end to have some model that captures different scales of description. I'm very content with the idea that what we do as scientists is basically describing adequately systems, if possible with some predictions. And that is where I think the level of quantitative analysis that we can do today and the opportunities to collaborate with physicists allows you to go from a very quantitative phenomenology to some predictive abilities. And it sounds a little bit like science fiction to be predictive, but I think that for a lot of what we do, and essentially nearly everything of what we do in our lab, it comes at a stage where the number of information and the sort of nonlinear interactions between components, feedbacks, makes it very hard, at least to my brain, to get a sensible understanding of what is going on and to predict particular phenotypes before we do experiments. So maybe this is a little word for the people who are in the course just arriving now. I think that it's a fantastic era now in developmental biology when, indeed, through these collaborations that we've been involved in through the courses, it becomes possible to actually make sense of your hypothesis at the beginning of your work rather than at the end when we draw in papers sort of cartoon models where we don't actually test the rigorosity, the consistency of what we propose, because we can't be even either predictive, or we haven't actually made the work of actually setting up the rules of interactions that we come up with at the end of our analysis, whether through genetics or biochemistry, and try to see what comes out of it. Does it make sense given what we know? So this is sort of a long preamble on my own introduction to, I think it's an important aspect of our research today, and that is particularly true when you address 2 problems where there are different feedbacks, is addressing interaction between different scales. And I think from different talks and what I will tell you today, you'll have a picture of a sense of how these different scales might be important. We can sort of work the problem from the subcellular scale all the way to the tissue scale, but you could also ask questions about intermediate scales and interaction between components. We'll see that. And another important challenge is obviously the interactions between biochemistry and mechanics. There's a great deal of biochemistry which controls many things, one of which is actually the forces that are produced in cells, how they are transmitted between cells. And this creates a movement of material which itself could actually feedback on the spatial temporal regulation of biochemistry. And so these feedbacks between biochemistry and mechanics are truly very important. And this is a place where I believe that having good quantitative data is important, and having some kind of framework to model what's going on is extremely helpful to make sure that what you conclude is consistent with the data and making sense. So this is an illustration of what I said the other day. Development can be viewed as an interaction between an information flow at different scales and mechanical operations which can produce more or less complex shapes. In development, as we'll see, there are more than 2 mechanical operations, and the information is probably more complex than the one that flows through the formation of origami. That allows me to illustrate the fact that during my talk today, I will try to touch upon at the end of my talk about some information that we think is important to organize developmental processes, morphogenetic processes. But we'll talk for most a great deal about mechanical operations. And we'll try to see how seemingly different processes actually emerge from fundamental common properties at the cellular level, which nonetheless produce very different outcomes. I didn't have any coffee, so I'm a bit slow to start. Sorry about that. I'm sure you all appreciate that. So we begin with the tissue, which we like to think is essentially two-dimensional, although, as we'll see, it is going to be a piece on an embryo, and so it's part of an envelope, if you want, that is spherical or ellipsoidal, doesn't matter. And thinking about this deformation, developmental biology has come up with two general classes of deformations, one of which you also find if it was a plasma membrane, you can form an invagination, how you make two tissues out of one. There are many examples in animals, whether the mesoderm or endoderm invaginations in invertebrates, all the neurotube closures in vertebrates. We'll talk about that, and Eric talked about that in some detail. And the other one is an extension of the tissue, which is essentially what happens when you form your anterior-posterior axis as an embryo, and that is true of nearly all organisms. And so we'll see during the course of my talk. So my talk will be pretty much a comparison and contrast between the different processes and try to see what is happening at some point here that explains this sort of bifurcation in processes, in developmental processes. And try to understand it from the standpoint of the mechanics going on in the cell, as well as potentially, and that will be sort of the end of my talk, with regards to the information that is actually instructing these cells as to how they should organize their mechanics. And that potentially provides a working model as to how this bifurcation occurs during normal development. So I will talk about flies because this is where we do our research, and this is also a very great system where we can have a very detailed quantitative phenomenology of what the cells and tissues are doing. And this is also a place where you can actually study precisely these two morphogenetic processes that I mentioned, namely the extension of an epithelium, which would occur on the lateral flank of the embryo, sort of this green area. This is a fly embryo. You remember maybe from my first lecture a month ago, already a month. It's about 0.5 millimeter in length. It's surrounded by an envelope of epithelial cells. The lateral cells in green here are going to extend. We'll see that in a couple of minutes. So here we have a good model system to study how tissues extend, and we'll come back to that. And in the ventral part, the mesoderm, there you have this invagination process that is important for the formation of the mesoderm. So in the same embryo, every single embryo that you take will carry in a way that is completely invariant, not in the detail, but invariant in terms of the outcome, these two processes. So you can ask, how is it that cells that are born alike, for about 5 minutes you cannot distinguish a cell that is here or there, and at some point a group of cells will form a furrow, and other cells will produce and participate in an extension process. So I think it's a great system and a great question, and it's a great system to answer this great question. So just as a one sort of half a slide summary of what we know about invagination, I'm taking this example not from flies, but it would be about the same. This is from an ascidian embryo, which looks like a raspberry at this gastral stage. You could see the cells in the ventral side forming the endoderm, which are the precursor of the endoderm, which constrict the apical surface, and that is part of the process that leads to its deep invagination. And that is dependent upon the apical recruitment of the force generation network of actin, crossing actin filaments with myosin to motors, which produce this force, an apical force which you could quantify with ablation, so to say, or actually have relative measurements of its magnitude. And you could model it in terms of isotropic traction forces exerted on all the different edges or junctions of these epithelial cells. And this leads to an elastic deformation and eventually some kind of local furrowing until the point when you get a deep invagination. Many labs contributed this simple cartoon model from Maria Leptin, Eric Wishaus in flies, Ed Munro, Goldstein, C. elegans, Sidens, etc. So here we're trying to address now the process of tissue extension, and this is one of the earliest movies which we had. Here you have minutes and seconds. And it was known at the time that cells intercalate, as it was initially described in Ray Keller's lab in Xenopus, and Ken Irvine, who was there but has left, actually characterized the first sort of intercalary behavior in these cells using some other microscopy. And what was very apparent in these movies is that cells are actually flowing first ventrally, but that's because here you would have an invagination. But more dramatically, they would flow to the posterior, which is the right part of this movie. And you could fancy a model where there would be some kind of chemotaxis gradient that would attract the cells to the right. And this is completely wrong. There's not such a thing as a chemoattractant, which allows me to say that from just looking at motility and movement of cells, you can construct many models, all of which are not true. And it is very important to know what is the referential. This goes back to Olivier Pourquier's talk about gastrulation movements and actually subtracting movement of cells versus a matrix. And I think that a great deal of papers on so-called chemotaxis in gastrulation are completely wrong, because precisely they didn't look at the referential. There are many referentials that could be taken. And I will, in the next couple of slides, present to you a simple model of what underlies this actual global motility of cells in the embryo. It has nothing to do with chemoattractant whatsoever. So here, and I'm again summarizing because I partly introduced this, plus some other people talked about it, Pierre-François in particular. And if for some people there's some missing information, I'm happy to sort of elaborate a little bit upon it. But essentially, these cells are going to exchange neighbors. So, and I realized that I may want to draw something here. So if you take the cells in this tissue, these are these hexagonal cells. So you will have cell A, B, C, D, right? And so what you will observe after about 15 minutes is that the A cell A, B, C, D. So you can clearly see that the cells have indeed intercalated in the sense that they have exchanged neighbors. The A and C cells which were in contact no longer touch each other, and the B and D cells now have developed new contacts. This is looking at the sort of apical view on the cells. These are the junction, the contact between cells, and you can see that they have exchanged neighbors. Now, to really see and capture what is going on, the easiest is to actually look at the geometry of not the cells, but the interface between them, right? And you can appreciate that something very simple is going on, which is shown on this slide, namely that they go through this irreversible transformation of the geometry of contacts, where the contacts between immediately anterior-posterior neighbors disappear, producing these tetrads or higher order structures called rosettes. And then these two cells here, having equal contacts between each other, now develop new contacts at a perpendicular. There are two very important features in this simple cartoon. One is that it is irreversible in the wild type, as I mentioned. And there is a spatial correlate to this irreversibility in time, which is that the process is so-called planar polarized. So we've heard about planar polarity, which is this sort of very general, probably ubiquitous property of cells to actually read coordinates or sort of axis and polarity in a tissue, and using and reading this information to translate it into some kind of either differentiation marker, like making a hair, or orienting the spindle during cell division, to orient division, or here to orient the remodeling of the contact, which is another readout of that. So planar polarity is a very general property, and here is sort of another example of planar polarity, not in the sense of the chemical, planar cell polarity pathway, but in terms of the process going on. Yes?
So the yellow cross is like a transition state. I'm just wondering whether it's reversible from that point. I mean, is there...
So in principle, so I don't know there why it is, and I still don't have, despite quite a number of years of thinking and discussing about why is this configuration not, well, if you believe that thermodynamic models are making any sense, why it's not stable. It does not make, the tissue doesn't like to stay in this configuration. Well, so in principle, it would either go back or go there. This is symmetric from the standpoint of the geometry. But it isn't from the standpoint of the biology. And you could say that the very process that led the system to go from here to there would prevent it from coming back. And I will mention in a couple of seconds, shown on the left, that there is some differential tension going on there, which is responsible, or the differential Mies interdistribution, which is important for moving the system from this state to that state. So maybe this very same, tensile activity is making, or is acting against any tendency to come back. And I will even frame it more explicitly by saying that, at any contact, there are going to be the opposed action of two forces, a weighting force coming from adhesion, and a tensile force from actomyosin network. They have antagonistic activity. Despite balance of force, the system is basically moving gradually through this configuration, and the cells want to restore their contacts. They cannot come back because of the tensile activity. Ergo, they will go into this configuration. But Claudio, who gave a talk sometime 10 years ago, showed that actually you need tensile activity to actually make this transformation. So it's not as simple as that. Okay? But here I want to say that this is irreversible and planar polarized. And because all the many cells do the same business of intercalation in the same direction with the same global orientation with respect to the coordinates of the embryo, that results into some net movement of the center of mass of the cells, which collectively produce the extension of the tissue. That is...
So what happens if you block gastrulation or the ventral invagination? What happens here?
So if you use a FOG mutant, which blocks the mesoderm and the posterior midgut, intercalation continues. Now, and Claudio presented that because the tissue cannot sort of extend along toward the posterior. The gate, sort of the door of the room is closed, so the cells want to intercalate. They do intercalate, okay, but they cannot escape. So there are the cells remain for a longer time into this configuration.
But they still never go backwards?
No, they don't. They do intercalate. So you could, in some conditions which Claudio reported, you could uncouple extension from intercalation. Intercalation can occur in conditions where extension cannot occur because there's not enough space.
So if you focus on the T1 transition, what is known about what is happening on the basal sides? Do they just passively follow, or there is a lag?
In a nutshell, and probably I will elaborate upon that, apically, so we used to have a view where apically there's something very special which drives irreversible transformation like this, and basally you basically follow, like you follow in it. But it's not true. I will show that there is extensive fluctuations around some mean behavior everywhere. The difference is sort of a consistent bias in the directionality of these fluctuations apically versus basally. So basally is basically exploring all many conformations, while apically there is a global trend. Now, clearly, basally the cells would eventually follow to keep in a configuration that is not twisting the cells too much.
What is so you mentioned the word transition state. So what is the characteristic time to go from one stable configuration?
Shrinking a junction is typically a few minutes time scale. And it doesn't seem to vary too much. Even in processes where the frequency of T1s, which are for sure these events, are rare, when they occur, the speed is about the same, maybe reflecting some fundamental properties of actomyosin network. So this is dependent on polarized tensile activity. You can see here that myosin 2 is far from being uniformly distributed. You can find these sort of lines of distribution. And as Professor showed you, you can do ablation experiments showing you that, yes, indeed, tension is actually anisotropically distributed. It is greater along these edges than along the other ones. And you can even make models and show that if you implement this sort of polarized tensile activity using vertex models or the like, then you end up having intercalation, which is pretty much like what you see in a real embryo, suggesting that that's enough to explain intercalation. We'll definitely revisit this model and actually go into some greater understanding of it in a few slides. I wanted to mention to you that, yes, this is occurring in flies, but now as imaging in other organisms become more and more, becomes easier and easier, you can find a number of other occasions where similar, remodeling in epithelial cells occur. And in fact, if you think about it, if cells remain epithelials, if they have to intercalate, they have to go through T1s. So, there's nothing really mysterious there. Now, it turns out that this always is an active process. And here, to give you an example, this is a neurectoderm of the chick embryo. And if you look at the apical surface, which is this inside part in the neural tube, this is work from Masatoshi Takichi, you can find that the epithelial cells have some polarized distribution of actin and myosin 2. Here is phosphorylated. You also have these sort of lines of cables, and actually they track cells during this process, and they showed that, yes, indeed, they intercalate. These red edges tend to disappear, and you have a formation of perpendicular edges in blue. It's a little bit more disordered than what we have in flies, but I think it tells you that there's a number of different organisms in which this happens. Another beautiful example where this happens is a chick gastrulation. You have this epiblast, which is a two-dimensional layer of cells, epithelial cells, and they have these massive large-scale movements that are symmetric around some midline. And these movements are powered by intercalation events, powered by polarized distribution and regulation of myosin 2. So I think you could say that if it's true in chick and flies, it must be universal. And I think that we'll see more and more cases where intercalation are powered by polarized regulation of tensile activity at cell boundaries. Okay, so that leads me to this extension of this previous model that, spatial control over myosin 2 tensile activity has the ability to actually control different tissue-level outcomes. If myosin 2 is isotropically recruited apically, then there could be isotropic forces acting against the junctions, producing these elastic deformations of cell apical surface reduction and tissue invagination. If, however, myosin 2 is now concentrated along contacts between cells along a given axis, then the cells will dissipate the stress by T1 events, intercalation. This is images from a real embryo in which I painted myosin 2. And so clearly, the embryos must have a mechanism to control myosin 2 and thereby control the distribution of tensile forces. And yes, indeed, if you do ablation experiments and break apart the actomyosin network here or there, you have different outcomes. Here, there will be polarized, there will be isotropic relaxation kinetics around the point of ablation, which you can measure. And if you do it here, then there's going to be anisotropic relaxation kinetics, reflecting, indeed, that the myosin 2 pattern seems to correlate with the distribution of tensile forces in the cells. Right? Sure.
So you said in the beginning that it was basically a contribution of tensile forces. Now, what you're telling us is that the Western is not regulated really. The point of regulation is this. Is that what
that seems to be what. Well, as you will see, I don't want to undermine the role of adhesion. I think you could say, interesting, and I think I mentioned that in one of my talks, that you can find in the 90s sort of the culminating point of the belief that morphogenesis is controlled by adhesion forces. And you could go back from Alfreter to Steinberg and sort of goes at its apex in Takichi, 90s, the cadherins, all the immunoglobulins were identified. We knew the molecules. We knew what they could do. Their expression patterns was extremely interesting because it was not ubiquitous. There was some tissue-specific expression. Takichi would show that in the neurectoderm, when this tissue invaginates, yes, there are some special cadherins and cadherin plus other ones. Plus, we knew the sorting experiments were working with as little as 50% difference in cadherin levels. So the problem is that from the past 15 years of research or more, it is pretty clear that all the movement of cells, intercalation in particular, is actually mainly driven by spatial control over tensile activity. So the sort of fluid behavior of cells sorting between each other or intercalating is an active process controlled by, dependent upon myosin 2 spatial regulation. So of course, this is acting against the frictions of cells, which at a minimum must be dependent on cadherins. So that is the first level where cadherins must be important. The question remains whether playing around with levels of cadherins you could slow down or actually speed up a process. At least in this system, doesn't seem to be the case. Okay. But now we'll rescue the role of caterenes by saying that, wait a minute, caterenes are not just additional molecules. There are also, as we'll see in a few minutes, complexes that are coupled to atomizing systems, and Denise talked about that in her talk. And this very ability to transmit forces at the junctions, at the cell interface, is precisely what makes atomizing network able to deform cells. So you need caterenes for that. And the question is whether caterenes have some interesting spatial and temporal pattern, which, if not regulating adhesion per se and thereby controlling morphogenesis, might actually regulate the way by which tensor activity does its business. So this is sort of a summary to say that it is inadequate to believe in morphogenesis being controlled by sorting phenomena and energy of interface controlled by caterenes. I don't believe in that, and I think it's difficult to believe in that. However, tensor activity has a has a lead role, but the activity of atomizing networks depends on caterene distributions, as we'll see. So at this point, we could stop working on German extension because we know a lot about it. But as I will, and you could view that all you need to know is understand how spatial control is being done. I would like now to spend some time actually delving into some subtle differences, some more details about how atomizing dynamics is occurring in this system. And as you will see, getting into these details allowed us to propose a working model as to how cells born alike become so different and do this business with that business. Okay? So to do so, I would like first to remind you of something which Eric Wishaus mentioned and talked about, which is that, in fact, these very cells which undergo apical constriction in the mesoderm also have an interesting property of having pulsatile atomizing contractility. This is true, actually, of many cells, even non-epithelial cells. In the C. elegans cortex, you have the same. Actually, the first report of pulsatility is not here, but actually from a paper from Embryo in C. elegans, where he characterized something we didn't call pulses, but were essentially pulses, where you see myosin 2 aggregating into larger structures, which have a half-life about 100 seconds, and few cycles, 3-4 cycles of atomizing contractility produce incremental reduction in the surface of the cells. So yes, indeed, there is a global trend. In that sense, it is globally irreversible, but locally there is reversibility. Okay? But there is a bias, you see. The relaxation is never as much, or in general is never as much as the steps of shrinkage. Okay? So here, this led the authors to propose that there is some kind of a ratchet mechanism. It doesn't matter whether you strictly believe that it's a perfect ratchet or not. The fact is that there are fluctuations, very important, that there is a bias in these fluctuations, and that the system has an ability to sort of lock the system into some configuration for some time. Okay? Sort of keep memory of the cell shape so that eventually the process is irreversible. And it's working pretty well. Within a matter of about 10 minutes, everything is invaginated. So it's not a sort of bad process where you have an inefficient deformations with relaxation. Okay? Yeah?
Data at the single cell? Or like the
yes.
Single cell? Okay.
These are single cells. And here you have the derivatives, rates. Okay? Actually, if you look at the cells yourself, it takes a bit of time to come up with a simple notion because it's, like always, papers, it's completely correct. But, cells do many things. And, the apical surface is not uniform. I mean, we've asked a student to actually even do some analysis ignoring what's going on. It might have taken them some time to come up with a simple kind of description. I think it's important to realize that, papers show one example, even in the end show averages, show good statistics. But if you look at the data and ask yourself what's going on, it can take you, a few days, if not more, to figure out what's going on. Okay? But as far as we've looked at, it's completely accurate, what is stated in this paper. Now, they model something like we all do. It contracts and then stabilizes and contracts and stabilizes. Some people question, what do you mean by stabilization? And we'll come back to that because, as I will show you now in the next few minutes, in German extension, during intercalation, you have a similar process of phases of deformation, which I would call constriction, and phases of stabilization. But no, they do not occur in the same areas of the cells, which has some very important implications. So this is the work of Matteo when he was back in the lab. At the time, he was working really hard. And there was no surfing there, so he could work. No, I'm kidding. So you could look at the cells so essentially, I think everything in our lab actually moves through steps, which depending on how better you can image things. And this is an important stage where with a 100x 1.4 NA objective, spinning this confocal, you could zoom in on the cells and look at things which we sort of deliberately ignored for a number of years, namely that myosin 2, sure enough, is polarized at junctions, but there's a lot of stuff going on in the apical surface, which we, it was opportune for us to ignore it because it was very hard to make sense of it to start with. But at some point, it was important to actually try to understand what's going on. And so these, you see speckles, they move about. They are diffraction limited. Probably these are mini filaments of 12 molecules of myosin 2. FCS measurements are consistent with that notion. Then they aggregate into larger structures about micron size, and they also have lateral dynamics. We'll come back to that. They work on a network of actin filaments, which is shown here with a marker against using a eutrophin actin binding domain. Clearly see, when you see these movies, you realize that it's not adequate to think of intercalation in terms of what happens just at the boundaries. It's a gigantic viscoelastic network where junctions are merely points of cross-linking between two pieces of the same network, you could view, right? And so we have to now establish a model based on these very phenomena. And so this is a medial actomyosin pool, to call it simply, and the junctional myosin 2 pool. And this is a typical example in a single cell of the kinds of dynamics of medial, red myosin 2, junctional myosin 2 in green, and the length of this junction, which is the morphogenetic output of the cells when they enter intercalation. This junction, you remember, shrinks, and it does so, from 4 microns down to 0, all the way down to this 4-way vertex configuration until something else happens. And this shrinking, which requires energy, requires myosin 2, actually, you find that there are two interesting dynamics of myosin 2 going on there. The myosin 2 at the junction, in green, increases over time. And this we knew for quite some years. It does so in steps, right? And you can also see that the medial myosin 2 pool also is pulsatile. On this very example, it looks like the amplitude increases, and it does increase here. But if you look at many, there's no global trend of increase in amplitude of the myosin 2 pulses. The frequency is typically in the range, or the period is in the range of about 100 seconds. We'll come back to that. And what you could see here is that any shrinkage of the junction sort of coincides with two nested pulses of medial and junctional myosin 2. There again, you have a shrinkage coming on with myosin 2 pulse, and then green stuff coming in at the junctions, and the last one, the 2 here. Now you can look at many, many such traces, do cross-correlation analysis. Matteo came up with a very surprising result, which was that the maximum rate of shrinkage here is occurring about 9 seconds before the maximum of rise of myosin 2 at the junction. So this very myosin 2 pool, which we proposed was so important for intercalation, and which, by the way, is very important for intercalation, okay, don't misunderstand me, cannot be the whole part of the business, because in a way its rate of accumulation is occurring too late, at least for a simple model of how this deformation would occur. However, there was always a slightly preceding medial myosin pool, and indeed if you cross-correlated these dynamics, taking the derivatives, then these two dynamics would be perfectly coincident, suggesting, I see two hands calling, suggesting a hypothesis using correlations. Correlations are very useful because here they allow you to propose a temporal sequence of events from otherwise extremely messy data. Not messy because you are messy when you acquire them, but because they are just complicated, as you see. Yes?
So can you just clarify the y-axis?
Sorry, the y-axis, there's 2y, right.
So there's only a 2% change?
Right.
In the total medium?
Right, so here it's very important to see how the quantifications. The quantification is that this all down to background levels, for instance. So here in these typical systems, whether cadherins or myosin 2, whatever, you have more than 80% background. So if you don't separate the background, okay, the difference look very minute because you have 80% or 90% plus something. See? So now if you do background, then it can become twofold, right? But now if you do the statistics, it's very reproducible. So fine, it is very minute but significant, and I explain to you why. Yes?
What about the area of the cells? Do they?
They fluctuate, but globally the area, they fluctuate a bit. Yeah, you could view, as we'll come with, that intercalation or a step of shrinkage is a bit like a polarized apical surface reduction. But the important thing is not the area. The cell doesn't care because globally the area is going to fluctuate but remain constant, while the junction, is shrinking. Okay?
One thing to cause another thing, it has to come first.
Yes,
So,
I think we all agree on that.
So, I mean,
Is there anyone objecting to that?
Is what you're showing us that the junctional myosin accumulation is not causing the constriction?
Well, it depends what you call constriction. The constriction in the sense of a step of shrinkage, it cannot, I would argue. However, for the persistence, it would. And I sort of preview what we propose is that this green bit is required for this stasis of the system, okay, for lowering the probability of relaxing. And in fact, I'll draw it later on. While here, this red thing is required for the shrinkage. So this is a working model, correlation, hypothesis. We have to revisit and say, well, maybe red stuff shrinks, green stuff stabilizes. Now we can use ablation experiments where we can precisely make a 2 millisecond pulse that makes a diffraction-limited ablation spot here, leads to the collapse of this pulse, and as I'll show, well, I will not show, but as Mathieu has shown, this is causing a failure to intercalate. And the junctional myosin is also required for that. But, yes?
No, I'm just thinking that what you told us was you pooled a lot of data and did statistics and correlation, but actually strictly it has to be true every time. I mean, in each individual cell, whatever's causing the other thing has to happen first, not just statistically over the large.
Which is exactly what happens, by the way. Yes, precisely. So when you do this cross correlation, you have to do it in the same cell. Otherwise, yeah.
So I just ask you again because I have the same problem in my work that the normalized intensity of myosin has this small variations like 0.04 in my case. So how you can be sure that this is not a noise? Do you have like a control situation that you, for example?
So that was what I was saying before. The myosin 2 that you see is something that you filter. This is coming to how we process images. But if you quantify, the seemingly haze of myosin 2 amounts for more than 80% of the signal. And that's true of a lot of quantification. So if you make an apical area or junctional measurement, okay, and we know this 80% because we measured it. So myosin 2 is in a cytoplasmic pool. This is a regulatory light chain exchanges with the network. Now the one that matters, the one that powers force, is the one that is in the network. So you need to find a way of quantifying this network. So there's 2 ways of doing it. One is by subtracting background, which is not background like noise. It is actually cytoplasmic myosin 2 in this case, and thus you can very precisely measure it.
This is exactly your normalization here. This is how you normalize your data.
No, here this is not with subtraction.
So what is this on the y-axis now?
So here this is total myosin 2 measured, the intensity in an area. Okay? So if you have 80 or 90% of something and you have some 10% or 20% at most, that is the one that you care about. If this is changing twofold, and if you make the ratio analysis, okay, it will not amount to a twofold change. But if you subtract background, then it will have a twofold difference. It's the same as the planar polarity of myosin 2. If you subtract background, you can measure a more than twofold difference. And if you do not subtract background, then it's going to be a few percent only.
But the background then has to be like always constant, shouldn't it?
Yes, and the cytoplasmic myosin 2 is constant, because this is a pool of the cytoplasmic myosin 2. For any single experiment, depending on what you measure, this background will have to be assessed differently because it will be different.
But do you think that if you, for example, look at the junction that doesn't change at all, doesn't change the length, you do the same quantification, you think that you have like different numbers than 0.04, 0.04?
We can go back and discuss this. I propose not to have a 15-minute discussion on how you set background. This is very important, and I appreciate it for your work. It's important. But I can tell you that what is true in any single experiment is that background could be the cytoplasmic level of the protein, could be the membrane level that is not in complexes for cadherins, for instance. And it is very important to know what you want to measure. The complex cadherins is going to be brighter, has to be filtered out. And the cytoplasmic myosin 2, does not power force. Yeah, and it amounts for most of the signal. So if you don't take care of that...
I agree with you. I'm just saying that people are always like,
right.
When I present my data, I have exactly the same data.
We'll discuss. I'm happy to do it. Yes.
Thank you.
I wanted to understand your answer to the question about the area.
Yeah.
So as the shrinking junction decreases its length, the area stays more or less constant, right? So the neighboring junctions, they increase in length. So what is happening?
Yes, when something shrinks, someone else yields length. Yes.
And the area stays roughly constant.
Roughly constant.
Okay, just.
There was a question from black holes.
Yes, thank you. If you could back up.
Yes.
So in this particular case,
Yeah.
you've got two cells
Yeah.
that are bordering the junction of
Right.
the question. And so does this mean that the medial myosin in both vary in concert? And if so, do they signal each other?
Very good question. I will come back to that. Very important aspect. You could already see that, this, because an interface is shared by two cells, you might think of how their activity is synchronized or not synchronized. I'll try to come back to that at some level. Yeah.
I was just wondering about the horizontal junctions. Are they going, because the shrinkage is going to be between this one and the other one. Have you also quantified how they are sort of changing the levels? Are they staying constant during this process, or are they also going down?
You talk these ones?
Yeah.
Okay. As this shrinks, this yields a bit of length.
No, the
perimeter is more or less constant.
No, myosin intensity.
What about myosin?
Oh, myosin intensity. Well, so myosin 2 is not accumulating there. It's not stabilized there. And I gave you the preview. There is a relationship between here and there, and that's shown in the next slide. Okay. There are not just pulses and junctional myosin 2. It turns out that the two are intimately linked because these medial myosin 2 pools actually flow toward the junctions. So as they do the business of contracting, what is a pulse? It's basically is a aggregation of motors acting against actin filaments and exerting tension in the network. It turned out that as they do so, they seem to be attracted toward any of the vertical junctions that is going either to the left or to the right. Here's an example of PIV on this actomycin, and actually flows in this case to the left, and will produce an increment of shrinkage to the left. Okay, and we'll see that in the next round of pulse, it would flow to the right and then to the left. There's going to be some oscillatory behavior there, which is not captured really in this particular image, but we'll come back in a second. So the sort of summary of what I've told you so far is that the deformation comes from two different actomycin pools. One is medial apical, produces deformation in a way that is in steps through the pulsatile activity, and is polarized because it flows to any of the junctions, whether to the posterior, anterior, or posterior, and in alternance. Okay, I'm showing one step. This produces an increment of shrinkage. Now, if there was nothing going on at the junctions, that would relax. And indeed, these flows are definitely not 100% exist. Sometimes they flow to this junction. They will produce a deformation. But because there's nothing here to stabilize the junctions, in particular, there's no myosin 2 that accumulates there, the junction will relax, so you cancel what you've done, like Sisyphus. While here, myosin 2, there's a mechanism that actually stabilizes myosin 2 there, which is what we began with in terms of the analysis, and that actually keeps and locks the system in this configuration. So you see there are two nested polarities. One in the network that produces the deformation through the planar polarity of these flows, and there is one in the stabilization of these cell shapes. Here is sort of a working or sort of a hypothesis. We have no data to support that. But you might think that if there are different mechanisms to control the polarities of these flows and the stabilizing system, you could tolerate bigger error rates on each of these systems because the error rate would multiply from, say, 10% to 1%. And indeed, we know from the measurements that this polarity is very clear, but it's far from being 100%. Flows can go in some of these transverse junctions, but they will be, so to say, punished because nothing will stabilize their work. nothing, the junction will not be polarized. So, it is a fact that there are two different polarities, and that they are both required for the process of intercalation. So then is a question, if you didn't have that, you would produce deformations, and even if they were polarized, in the end, you will have to do the business again. Now, it turns out that during intercalation, you just need 3 pulses on average to produce a complete shrinkage, right? So you don't have to do it 3 times, and that's it. And now, of course, you can see, and I mentioned to you that the flow will be on the left one stage, but it will have to be on the right at some point. But now one junction is shared by 2 cells, so you immediately try to think, well, there must be some interaction between these vectors at the tissue level, and we'll come back to that in a few minutes, okay? Because this is a very local explanation of the process, okay? But now we have to sort of reconstruct mentally what happens at the tissue level.
Does this help us with the fact that when we look at the embryo, the junctions are not all perfectly up and down?
Yes,
sometimes
sure.
They're a little bit
right.
off, and does that help fix that too?
I think that if you want to predict what's going on, you need to look at more than the vertical sort of junctional myosynth. You need to look at the deformations. You can produce conditions in which there is hardly any medial myosin 2, despite a very good planar polarity of myosin to the junctions. Intercalation is very, very strongly reduced. So you need the 2 systems. Okay. With regard to the angles, I told you about vertical junctions. Is there any objective criterion for that? Well, the embryo is curved. You can actually measure a local vertical thing in your frame of reference. And indeed, there is an empirical rule. Within a certain degree from this vertical, you can define an angle in which, if the junction enters, then myosin 2 would accumulate. We don't know what this reflects, but presumably something about how the polarity, the information is now produced and spread in the cells to actually eventually control actomyosin, myosin 2 activation. Okay? So at this point, and sort of comparing and contrasting the work of the Wisha's lab and our work, we would propose a working model in which the cells are born alike. They have adherins, they stick to the neighbors, they have actomyosin that forms this, has intrinsic ability to pulse. I'm not going back to why they pulse, how they pulse. We've discussed in different talks how this could emerge in these networks. The fact is that they do, the period is about 100 seconds, but within a few minutes, the cells in the ventral part of the embryo, the pulse would not flow. They would remain sort of medial and apical, exerting thereby traction forces that are isotropic on the cell contacts. This produces only geometric deformations, which you could say are purely elastic deformations, by shrinking all the junctions a little bit. And eventually, maybe at the tissue level, there is some kind of buckling instability that is what is necessary for the onset of the process. And in fact, to some extent, a way of reading at the data that Eric Wish has presented us on this sort of convergence of actomyosin contractility is at the tissue level something not completely different from that view. Okay? I'm not going to try to fit in Eric's data in this context. He showed that in a way you don't need cells to have the tissue level, but clearly cells exist and they do their business at the tissue level through cells. So, but the very unique things of the ventral lateral cell is that there the pulses are sort of attracted, as I say, attracted to junctions, either the left or the right. And by doing so, they polarize the deformation by shrinking a bit of the junctions to the left or to the right. And by doing it 3 times, the junction is completely disappeared. And that dissipates the stress by intercalation and producing tissue extension. So we really need to now we are a few seconds. Before we were 1 minute after the Big Bang, now we are a few seconds. We try to see, well, there's something in these cells which make them able or not to flow or to flow actomyosin. And we'd like to understand that. Yes?
So now you sort of very clearly introduced polarity. So
right.
Flow has a direction as opposed to anisotropy.
Right.
So do they all flow in the same same direction?
So this is, I think, another formulation of your question, and I've decided to sneak in data on that. I'm sorry, this is going to be not as organized as I wish it, but I will do it now. I've squeezed in some data because I felt that it would be appropriate to talk about that. So I think, like always, in biology, polarity is very important, okay? And you can always conceptually break it down into 2 sub-questions. How do you produce a vector? So what produces a flow? And second, how do you orient these many vectors with respect to some global coordinates if there is such a thing as a global organization. And this is something with Jeff being here, and we heard it from other people before. In planar cell polarity, you have these two main questions. So to break a symmetry, it's very easy. You can have some negative feedback in the system. How you orient? Well, you'd like to think there's some kind of large-scale information system that actually buys this intrinsically, unstable system in a given direction. Okay. So I will not talk about specific information there. I will talk about mechanics, because we know a lot about how you produce a flow in actomizing networks. Different people talked about that. And we'll try to see how we have to sort of adapt this notion in the context of intercalation to try to see how it might work. So we begin with simple and what's known in other systems. If you think of a C. elegans embryo, these are images from Ed Munro's lab. Here's actin, here's myosin 2. This is a large cell about 50 microns in length. So any of our fly cell would be a bit smaller, but these actomyosin structures that are pulsatile are in size quite similar to what we find in our fly epithelial cells. Okay, so locally it looks the same, but this is a single cell, right? This is the same as, to some extent, the non-cellularized embryo that Eric Wishaus talked us about. This slam dunk mutant. It's the same sort of network. Now it flows from the posterior to the anterior, and that is essential for the polarization of the C. elegans embryo. And it was clearly shown, and I refer you in particular to work from Stefan Grill, who unfortunately is not coming, but showing that all you need is a gradient of contractility in this network to actually cause the flow of this network. And this property is actually, this system is actually self-sustained because the flow itself will steepen the gradients, thereby sustaining the flow in a way that is sort of permanent. Okay. The same sort of thing can be observed to some extent, and depending on cell type during cytokinesis, where on top of the local regulation over myosin 2 phosphorylation at the equator, which produces this constriction, you also have material flowing from the poles toward the midline in a way that is now symmetric. And there again, this flow of actomyosin is believed to be controlled by a gradient of contractility, which sort of sustains the flow irreversibly.
Does the flow here feed back on the gradient of myosin contractility?
Well, there are different.
In the worm?
Well, there are different feedbacks. One of which is that you transport myosin 2 from one region to another. So there's a source, okay, and you concentrate it through the flow. So clearly, you expect to have some, unless there is, and there is actually some dissociation of myosin 2 as well. So it's not that it's going to be like this, and then all of it is moved. The system is sort of stationary also because of some, depending on the flow speed, the advection flow speed, and the association dissociation of myosin 2. All of that can be modeled. But I'm saying that the system will naturally sort of self-sustain. Right?
And it self-sustains because it's like a driven cavity flow where you drive.
It's self-sustained because the gradient sort of remains. And now you could add some more complex feedbacks by the transport of the very molecule that activate myosin 2 activity, like Rho kinase or other molecules. That's a more complicated version of the same concept, that the system is self-sustained because the gradient is sort of maintained somehow. Now, that is in single cells. Now, we are trying to answer in cells that are single cells, but are connecting with the neighbors. They are contacting the neighbors, and the question is what's going on there. So,
yeah. I'm sorry, but I don't really understand why this cell shape does not change if you have a flow of myosin 2. Should it contract on?
Well, if you look at the local shape of the cortex, it is actually not smooth. There are some kind of bulges, and I think this sort of is caused by the local heterogeneity in myosin 2. Now I was talking about the large scale. I'm smoothing these irregularities, and actually Stefan Grill in their model, they don't model that.
Okay.
They have a very simple sort of coarse-grained model of the material moving from posterior to anterior. And if you want to know more about cell shape and that, you should ask them. Okay, so here they are single cells, and we want to know in intercalation. Well, clearly, in epithelial cell, the apical actomyosin network, and you may actually remember, can I go back 1 minute to a slide that I showed before? If you look at these movies, you can clearly see that, this actomyosin network, these are the boundaries of cells. So you might think, well, the contractile actomyosin network is going to exert traction forces on these edges. The question is, given the length scale of the the length of the cells and the length scale over which the elastic behavior might operate, which no one clearly measured, but at least if we have the same figures than the C. elegans embryo, in this case, viscosity dominates, so you have long-term gradients, long-range gradients, and therefore here we are much smaller. So you might think naturally that this contractile network would pull against the junctions, and immediately you have to consider, well, then the boundaries of cells must have a major impact on the very dynamics of this contractile system. And that is what I wish to get into. So just as a little preamble, in these epithelial cells, look here, greens are cadherins. These are Pontic structures, the structure of which is being heavily controlled, and Pierre-François talked about that. And you have also actin. What's not shown there is a medial apical actin. Well, because for one thing, at the time we didn't know that it existed. And if you do simple phalloid instant, you don't see it, or because it's a very fragile network. Fixation is not good for it. The important thing is that this actomyosin network is connected to these cadherins by cocktails of molecules, essentially alpha-catenin being the main molecules. There's been a controversy that lasted about 6 years about whether it's a single molecules or many molecules. Alpha-cat is the molecule that matters here in any organisms, vertebrates or invertebrates. And so we'll play around with this molecule. If you knock down alpha-catenin, and this is work from Romain here, who, when he was in the group, you do a very simple experiment. You go back to the default state, so to say, by removing junctions. So in the wild type, I mentioned that flows go left or right. If you now remove junctions, the cells round up. They don't know that they have neighbors. They behave like small C. elegans embryos, or like cells that would divide. They would naturally have flows of myosin II, because it turns out that apically, for some reason that we don't understand, myosin II concentrates, and you will have permanent flow of stuff toward this apical site of enrichment of myosin II. And you can see here the movie. It's infinite because it's looped, but if you look for quite a number of minutes, it is indeed very striking that this is sort of the default state. This is a cell in the germ band that would normally intercalate. You remove junctions, and flows now goes toward one single spot. So the presence of junctions redirect flows to the periphery.
Can they cellularize normally, or they don't cellularize?
They have cellularized normally. Yes. Yes?
I mean, why are they still here, and why is there still a polarity? I mean,
why are they still here?
Yeah.
Because they are sandwiched between the vitelline membrane and yolk, and they are good there for some time. Right. So they remain there for quite some time.
And where does the material go?
Well, this is the same as the C. elegans, so you can model it different ways, but simple ways, you have a little box. There you will have, advection flows, whatever polarity might have, a certain speed, which brings more myosin II in it. But you also have association-dissociation from the cytoplasmic pool. Okay? And depending on the balance of these three things, it can be steady state, it could be, you could increase the concentration. So here, there's dissociation going on, and in such a way that in the end the system is sustained. Okay?
This point that remains, is it polarized also?
Sorry, this? No.
It's everywhere.
So it tends to go apical. That's the only sign of polarity that we have. So these cells still know that they have a polarity. Quite interesting. What's going on there that is telling them, well, put it here rather than anywhere randomly? Boris?
What happens to cadherin when you disconnect it from actin?
No cadherins.
So no cadherin.
Yes, and if you remove cadherins, same result. It's the same result whether you remove cadherins with RNAi or alpha-catenin completely. If you remove a little bit of alpha-catenin so that you actually compromise the strength of the interaction with the neighbors, but you leave enough cadherin complexes, then flows exist. Now they have a remarkable feature. They are randomized. They are in the plane. They occur. You can track them. You can measure their directionality. You have no net bias, right? And so boundaries, this is a very crude statement, Boundaries attract flows. I think you understand what I mean by that. It means that the boundary conditions, matter clearly for what happened in these cells. And these boundaries actually are cadherin complexes. Now, cadherin complexes are structures that are symmetric. They are shared by two cells. There are these glue systems that share two cells. So whenever you have a complex between two cells, any cell on the left or on the right will see it, and will come to a little potential puzzle there.
So can I ask you
Yeah.
how the network is still at the membrane? What is holding it at the membrane? Because it's the form of the membrane.
It's like any cell. You know, this is what I call the default state. You know, they have this cortical actomycin. Many people talked about it. There's a lot of biology on that. I pretend that's all that we know, although we probably don't know all the details of it. I'm just saying that on top of these ways of understanding flows, here is a case where you have to consider boundaries, junctions. Okay? Because there have been many, many papers on explaining flows in single cells. And here we're trying to understand flows in sheets of cells connected by contacts. I'm just adding this little point, which is essential here to understand what's going on. So we know what happens when you have no coherence. Something interesting, but not really important, not really doing much for tissue movements. Now, you want to say, well, now, if I did a simple experiment where I had differential level of coherence, if I introduced asymmetries in coherence in a tissue, well, the expectation is that, I have these isotropically contracting systems, and of course, balance of force, coherence are there. There is connection between the actomycin system and these complexes. Therefore, the contraction of the network exerts traction force on the coherent complex. And if there was just, so Gedanken experiment, just coherence there will be a net movement of the flows toward the points of high coherence. Okay? So you can do experiments, and I will not go into all of these experiments because the time is running. But you can do it different ways. I will show you just one. But clearly, in all these manipulations, any asymmetry in coherence that you generate produces a direction of flows towards the high points of coherence. I'm showing you one where, if you do RNAi to E-cadherin, or alpha-catenin, actually, you can produce some patchy disappearance of E-cadherin. Okay? It's not something reproducible in the sense that, you don't know where and when this will happen. But that happens because RNAi is not perfectly efficient everywhere. And you look at regions where there is seemingly normal levels of coherence, and others where there is none. Okay? And you look at the interface, and you look at myosin dynamics. This is a PIV vector map. It clearly goes strikingly towards the points of high coherence. Okay? I'm happy to go through all the data later on. Romain is here. He did the experiments. I want to go into this result that we arrive at the conclusion that, and I'm just going there, if you have a step, an asymmetry in coherence, you attract flows. Romain did another experiment where he overexpressed E-cadherin at higher levels in the cells. That caused a situation where actually flows tend to be not completely blocked, but strongly reduced, because essentially actomycin contractility is going on, is exerting traction forces on all the many junctions, sort of to say equally on average, and therefore it doesn't know where to go, and it's sort of stuck in the middle. So that is, by the way, what we think is going on in the mesoderm when you have apical constriction. And indeed, conditions where flows are inhibited, like here, produce a mild, detectable apical constriction, right? So I would like now to yes?
So if I remember correctly, you see E-cadherin accumulate on the horizontal junctions that are being made?
Right.
Okay, you're about to talk about that?
I'm about to talk about that. So here I sort of engineered the system to see what it can do and to see whether in particular, although coherence is important, can I actually predict or make a model or working model of how coherent would have to be? So that's a question now. How would you design coherent levels to actually produce what actomycin is doing, which is that it's flowing with a polarity? Okay? So we're going to play a little play game, if you don't mind, because I think this is, you might arrive at what Romain found. So we are going to think in one dimension, and I will have, maybe someone can switch on the light as I'm drawing this. This is here.
I saw it. The board's up.
Right. So I'll make it 3 versions. And the last one. Okay, so what you know is that flows have this axis, AP. Okay, So you also know, and I've showed you some data, but everything is clearly consistent with this notion that high cadarians attract flows because of the coupling with the actomizing network. So how would you put cadarians to actually attract flows? Well, you might think, simple, I'm going to put more cadarian here. Okay. And of course, that is a problem because you have some, medial actomizing there, and it's going to be exattraction forces to some extent here. These are the traction forces, and on average that would be symmetric. I'm thinking about, not thinking about any sort of noise. I'm thinking about medial, average level. So I don't want that. So let me now just, because the system might be trapped. And I showed you data that indeed when the Romain has high levels of E-cadarian all around, indeed you find that the flow is reduced. Flow speed is reduced. And you could also show by ablation that the relaxation recall velocity is increased as a function when you have more cadarians and decreased when you have less cadarians. So all of this is perfectly consistent. So, well, what you might want to do is say, okay, simple, I just skip one junction. So here we are into this play game of asking what happens at the tissue level. Well then, that's good. I will have flow here, flow here, flow here. So now this is a flow, okay, flow. These were the traction forces, okay, just the opposite. Well, the thing is that as the flow goes, this junction will shrink and shrink and shrink. This would go into a T1 swap. Same here, but there will be some winner and some junction that lose the game. So that cannot work because every single cell in the tissue does intercalate. So how do you do it?
You alternate.
Yes, so you alternate, which precisely, so you have to have a system that says, well, it's going to be the even-numbered cells and then the unnumbered cells. Sounds a bit too beautiful to be true. Well, it turns out that another way of phrasing it is say, well, as long as I have an asymmetry in cadarians, and I say that it's not a stable asymmetry, but it sort of alternates and fluctuates, as long as there is some kind of fluctuating asymmetries in the level of cadarians in any cells, in some unit of time, and if it is in par with the periodicity of the flows, I will attract with certain probability flows when they emerge, because they don't care, they just produce, and if they just read the boundaries, and if they have a little bit more of cadarians here than there, then I will go to the one with high. And then the next pulse comes, and of course, cadarians will be different. Okay. So I'm going to show you that this is what happens, and maybe I will go very rapidly through that. But essentially telling you that you have to invent a mechanism to actually control the fluctuations in Ecadarian asymmetries. And you would want to say in particular that these asymmetries are strong in this axis and low along that axis, which is exactly what you can measure. Yes, Boris?
I'm still puzzled by
the
difference between the two pictures. So here you say gradient polarized.
Yes.
So there what you show is not polarized but anisotropic.
Okay, we have to agree on terms. So let's clarify terms.
Another one is a tensor.
No, sure. I'm talking about vectors, okay? And I'm telling you that an asymmetry of Ecadarian is producing a vector of flow. And that is accurate.
Inside each cell.
Inside each cell.
But it's disordered from cell to cell.
Yes.
Some
so
go right,
if you want to think from one cell to another,
go left, and
absolutely.
on average, tissue level, the only thing that remains is anisotropy.
Right. So exactly. So if you look at many cells, so many cells plus over a long time window, where many events of flows have occurred, what you measure is what some people say axialization, which is that it's bipolar. It goes with equal probability on left to right. So what I'm trying to do at the moment is to go from this picture, which is the one we had, to the one that makes more sense in terms of the mechanics, and which Boris alludes to, which is understanding in terms of vector. I want to understand at a certain unit of time, typically the lifetime of a pulse, 120 seconds, what's going on. Now, on this time scale, we observe fluctuations in Ecadarians, which I will show you are correlated in time and space, and the amplitude actually is predictive of their orientation. Absolutely, yeah.
just want to clarify. So you're saying that inside each cell you have an internal feedback that makes it.
Well, we'll come to the feedback because this cannot explain everything. In particular, it could explain how flows go at one point, but at some point you need to recycle for the next round. And there, we don't know everything. For any positive, so phenomenologically, right. So phenomenologically, what I've told you is that there are pulsatile flows. Okay. This is time. And then these are also oscillating. Well, you might predict that they must oscillate if your hypothesis is right. And it is. Oscillating because they would go in any cell, left, then right, and the left, then right. And they do. Okay? And this will correlate with the fluctuations in the E-cadherin. Okay. Then you ask, are these two fluctuating or oscillating asymmetries, are they synchronized? Are they interacting with each other so that the system is actually self-organized? We think they are. That these symmetries have a trigger and they have an amplification that depends on flow. I will not have the time to go through all the data. I just wanted to actually introduce a problem, see how we think it's working. And if you want to know more about that, then we can discuss. Roma is here. We'll have all the data to discuss this. And we don't understand every single aspect of the problem, needless to say, especially in the feedback. But if you think about oscillations, you want to say, well, there has to be some negative feedback that recycles the system. We have some ideas as where this could come from, but we don't have any definite proof of that. So just some data on the oscillations of flows. This is what looks complicated, but this is in time, okay, crossing a cell from left to right. Okay, if flows go left and right, you might see some in a chirograph. So left movement, left, right, left, etc. So if you follow single cells, you do see that myosin 2, goes left and right and left and right. You can actually measure by PIV this leftward and rightward movement, see that it is actually have some kind of periodicity. You do cross-correlation analysis, show that the typical periodicity, is quite spread, but around 120 degrees on average. You can do it different ways with flow PIV analysis, also measuring the ratio of myosin 2 in regions close to where they arrive. This gives the same result. Okay.
The strong left bias here is
well, it's just a single case. Yeah, it's pretty difficult. It's when you see it's fine. You can measure. This is measuring many such events. Okay, but two pulses are not the same. They don't have this beautiful similar. I could find a perfect example. Yes.
You have this very nice autocorrelation.
Yes.
If you do that into adjacent cells.
We haven't. So that's going to typically doing analysis of actually trying to. You expect that so as you have more cadherins in one cell, and if in the same time window, two pulses will emerge in the same cells, they should be attracted. But now the pulses are anti-correlated between neighboring cells in general. So you will rarely see this occurring. It happens sometimes, but it's pretty rare. The rule is that flows are anti-correlated between neighbors.
That's what I'm asking. You have some data on this.
Yes. It's a very general trend that you don't find them correlated. Just like you find that the shrinkage.
Not correlated, or are they anti-correlated?
Anti-correlated. And this has been found by, in amniocerosa, the work of Damien Brunner and colleagues, Solon, did report the similar kind of anti-correlation. Just like you have anti-correlation in the shrinkage of the left and the right junctions, right? So I would just like to show you this. This is sort of a little cartoon to illustrate what I was drawing on the bone. This is looking at the cadherin intensity. We can do it again different ways. You subtract background or you don't. The amplitude of the friction is much higher when you remove the contribution of this membrane cadherin, which is not complexed and making this big puncta. This is a cadherin that transduces force because it's the one that is in these homophilic complexes. I'm referring to Pierre-François Stork. You have 80% of cadherin that is this free cadherin diffusing around, present, contributing most of the signal. Okay? And if you subtract this membrane and liganded cadherin, and you integrate cadherins or the whole thing, so you sum up the amount of cadherin complex able to transduce force, it's even higher amplitude and more telling. It doesn't matter how you do it. The result is the same, that you have some fluctuations, okay, along this axis. If you did the same measurement along that axis, and I don't think I have the slide, the amplitude is much lower. So the conclusion there is that, I'm going to simplify and arrive at the conclusion that the amplitude, the pulses, the fluctuation amplitude of E-cadherins, okay, which we measured, are spatially organized. They are stronger along the AP axis than along the DV axis. Okay, I'm sorry that I'm not going through all the data. That takes some time. Okay, I think you have the picture that is what we propose, and we think we have demonstrated. And by the way, this paper was just online, so I think the substrate to discuss it further is the idea that the way you can explain the directionality of flows in general and also locally in time through the vector orientation is by considering not sort of the mean level of cadherins, but rather fluctuations around some mean and spatial control over these fluctuation amplitudes in time. That explains the oscillatory flows of actomyosin and actually solve the puzzle of how do you want all cells actually doing the business of intercalation, given that what produces these deformations are actually vectors, which, if they were always in the same direction, would cause a problem in terms of having all the junctions shrinking.
What happens when you embed this in a two-dimensional? Is this sort of a one-dimensional argument?
Right.
Are there going to be fluctuations that occasionally you'll lose the one-dimensionality? There's no penalty for drifting, and then you'll, over long enough distances, you'll lose the information.
So I'll agree, and I think this is an interesting question. And I think this is exactly where, for this sort of arguments, we need to be aided by some kind of computational model that sort of aids us in predicting certain outcomes depending on explicit rules about interactions. You know, we know a number of facts from experience. We think we understand the process, and I think it makes sense. Okay, we present it with the most simple model that explains all the data. But there are some other problems, like, you know, how the two-dimensionally might be a problem or not. In fact, it is not. So what is it about the system that allows it not to be a problem? How do you can you explain from that the coupling between cells or not? There, you know, because of feedbacks in the systems, we need to be aided by such simulations of some sort. And this is ongoing things. And I think that things we don't understand are typically how do you recycle the system, right? So what I didn't tell you is that we know what we think trigger this instability. It is some polarized endocytosis of E-cadherin, right, which lowers the mean level of cadherins, okay, and we think creates an increased standard deviation around that mean. And the flow itself, we have data that say that actually steepen the gradient, that ahead of the flow, cadherin levels rise during flow, and conversely in the back. So the system has a positive feedback. But at some point, you have to have some negative feedback in the system. And we don't know where this comes from. It could be that E-cadherin is endocytosed or sheared or whatever. You lose cadherins. And we know that, indeed, when you block endocytosis of E-cadherin, okay, you block flow, actomyosin is stuck in the middle. We have an even better experiment, which is that when you do an endocytosis experiment inhibition, you block endocytosis of many proteins, one of which is E-cadherin. But we use an immunoglobulin against E-cadherin that increases the rate of endocytosis of E-cadherin, okay, and randomizes flows. So we have a chain of reaction that the removal of E-cadherin at a certain rate, controlled specifically by this immunoglobulin, can completely fool the system into losing the orientation of polarity. Okay. So I think I might have to stop, actually, because maybe I want to ask another question. So here I wanted to sort of address the problems of temporal dynamics, interplay between cadherins and actomyosin, and the importance of fluctuation around some mean. Yes?
Can I just ask a question before you move on? So the rate of junctional shrinkage isn't constant. Presumably it's some kind of distribution.
The rate of junctional
shrinkage at any given junction. Which of the parameters that you talked about in terms of the intensity of E-cadherin, the amplitude of these flows one way or the other, the actomyosin pulses, which of them correlates best with the rate of shrinkage?
The rate of myosin 2 flow or cadherins?
No. So the rate of junctional shrinkage.
Right.
You've talked about all these parameters that correlate and are involved in this process and drive junctional shrinkage. I'm wondering which of these parameters correlates best with the rate of junctional shrinkage. Is it the amplitude of these back and forth flows that you're talking about, or is it actually the local concentration of the absolute local concentration of the molecules in the site?
At the junction.
Yeah.
Flows, so if you think about myosin 2, the best correlation is flows, not junction, in terms of the rate of deformation. Now, you might think cadherins. Cadherins fluctuate. So what about correlating cadherin fluctuations and strain rates? Okay. We can discuss that.
Okay, I mean, I guess what I'm getting at is I'm wondering if it's the absolute level or it's actually the differential.
Oh, it's differential.
Between
it's differential. It's not absolute level.
Does this back and forth flow actually play a role?
Right. So I think you're asking for correlating asymmetries in cadherin fluctuations and strain rates. I don't think we've done this specific analysis. But you might think they would be correlating. Now the question is time delays. Whether it's, I would say the time delay would be different. I would imagine that it will come. As far as I remember, the myosin 2 flows highly correlate, but there is sort of a negative time delay. So there might be some time to actually remove cadherins, which then would better correlate in time. Here we're just speculating. I think here we haven't done the analysis. These are important questions, trying to disentangle. I think they will correlate, given all the pairwise correlations. The question is exact sequence of events. Time delays are ordered of a few seconds here, so we have to do the experiment correctly. I would like to finish by asking a question and bringing you a summary. I will not show the data. I'm happy to discuss them. I've proposed to you a model where basically spatial control over flows. Don't look at this slide. Spatial control over flows explain direction of deformation. If it's planar polarized, intercalation. If there's no flow, you do simple geometric deformations. Okay. But now I've told you that it's very important to stabilize cell shape. Okay. So you want also a mechanism to control that. Okay. And this is shown in the next slide. Well, you could hypothesize that irrespective of this pulsatile flow dynamics, very important for deformations. If there's no system to stabilize cell shape, which has a correlate, which is the accumulation of myosin 2. Remember that in the mesoderm, Eric Wischel showed that irrespective of pulsatility, the mean level of myosin 2 accumulates over time, and that correlates with the ability to stabilize the cell shape. Likewise, there is junctional accumulation of myosin 2 here, which is needed to stabilize the cell shape. We know it's correlating with and needed, because if you ablate that, you lose the cell shape and you relax completely. So that begs the question of what is special about this actomyosin network that actually conveys stabilization, and here the spatial control over that. Okay? And you could propose in particular that there would have to be something such as a stabilizing function, call it X, whatever, which we think actually works mostly through phosphorylation of myosin 2. I mean, Akangshi, who was there, has data that clearly show that when she hyperphosphorylates myosin 2, blocks its dephosphorylation, she locks the system into a state where it accumulates myosin 2 and would stabilize cell shape, right? Okay. So now this is going into a bit of development. Information has to be produced and talk to the cells to say, well, I want to accumulate myosin 2 here and there. Okay. So so the unexpected and quite remarkable results, and this is again simple genetic analysis. We found, people have been thinking, what is in particular in this case the planar cell polarity pathway that controls this process? We know what it's not. It's not what everybody else is working on. Frizzle, dishevel, prickle, and all the like. Fat Daxus, I don't think. Okay. As a little parenthesis, you might think that I don't think that flies have invented, a whole system that would be so unique. Maybe there is something common to many systems that is not yet apparent, either in the logic or into some part of the molecules. This system here, we don't pretend to have a complete understanding at all. we just found an important signaling pathway that turns out to affect that as much as it affects this. So it's a shared function, a shared pathway in the mesoderm and in the ectoderm, and it turns out to be required for the accumulation of myosin II in the mesoderm apically at the junctions in the ectoderm for intercalation is required for both processes, and it is none of the pathways that we know of, DPP that we heard about, WNT that we heard about, hedgehog that we heard about, all of that. It is a very general pathway for which there are thousands of receptors in humans, not maybe thousands, but hundreds. It's GPCR signaling. So I'm going to show you that a knockout of a GPCR, okay, 1 minute, Boris, and I stop. Yeah. So I think I will just go to the conclusion. I have many data to show you. Well, So we found a GPCR in a non-mutant. The pulsatility of actomyosin is normal in frequency, slightly reduced in amplitude. What is consistent in mesoderm and ectoderm is that the accumulation is gone, or strongly affected, and this has dramatic consequences because the cell deforms, does its business, but cannot stabilize anything, so it's called scissor field. So GPCR, so we call it, activates, we think, this pathway downstream, which is all this pathway that we know talks to myosin II and makes it active and happy and doing its business of contraction. This is planar polarized, this is planar polarized, this is planar polarized. We believe that Rho1 is planar polarized, but there's no evidence so far. So you'd like to imagine that GPCR, this GPCR, or some accessory proteins is also planar polarized, but we have no evidence there. And there has to be some X factor that is responsible for its activation. Okay? Now this X, we argue, would have to be regulated by genes which Ken Irvine has shown was essential for breaking the symmetry. These are these beautiful parallel genes that I mentioned. So you remember in my Blackboard seminar I mentioned the exquisite color identity of these cells. This is a way that you could see it. Each cell has a unique identity because it has a unique color when you label the proteins with antibodies, right? And so these differences along this axis is the template that actually is used by cells to actually produce an information that will break the symmetry. We don't know how, okay? If you make the cells all alike by some mutant that makes all the color the same, then you lose the planar polarity and intercalation is blocked. So we have to go from the orderly expression of these factors to some kind of flow of information that has some directionality to control myosin II activation.
Is it still forming ventral furrow?
In the GPCR mutant? Oh, in the... no, I believe it does, yes, I believe it does. So this interpreciation network is required for planar polarity. Twist and snail are required for the ventral, right? So here is some kind of a working model, having not shown you the data, so you may or not believe me. But this receptor is required for this myosin II accumulation. The striking thing is that you would argue there has to be some X component downstream of even rent, which are these parallel genes, to explain this junctional and maybe planar polarized myosin II. Okay? And in the mesoderm, so we know the ligand for this receptor. It's called FOG. Okay? So we argue that FOG is required for this medial apical accumulation, and some X ligand, which we don't know, would be required for this activity in the ectoderm. Now the remarkable thing is that if you put FOG everywhere, then these cells would be confused. Through the same receptor, they will think that they are in the mesoderm, and they will do this. This actually was shown earlier by Eric Wishaus, not quite in the way that you would like to do it today with the sort of time lapse and dynamics, but clearly this is what happens. So a same receptor, so this is sort of a working model. There's a conserved pathway. This is conserved in many animals. This is same GPCR in the ectoderm. Will control different outputs by spatial control over myosin II accumulation, and that seems to be somehow encoded in the kind of ligand which would activate these receptors. It will be FOG in the mesoderm, and we don't know what it is in the ectoderm.
The FOG overrides things.
Yes, somehow. And it's very weird, if you think of receptors and ligands, normally you have a ligand, receptors would be spatially segregated. Here we'd like to think that actually the ligand and its way of working encodes some spatial information, which is a bit unusual. But anyways.
Usually the default situation is the pair rule, and actually even first are expressed in the mesoderm.
Right.
And fog comes in to
right.
wipe that out.
Yes. You could think of different things. Right. I would like to mention that this GPCR is common to all the cells, is maternally loaded. Now there are some zygotically controlled GPCRs. There is one in the mesoderm, recently identified, but only in the mesoderm. And I believe we have evidence to support the idea that there are other GPCRs in the ectoderm. So
in a smog mutant, there is no ventral furrow, correct?
Well, no ventral furrow like in twist and snell, that cells get in after some long delay, but you strongly affect the rate of invagination. It's a bit like twist or fog mutants. I have to stop, I'm afraid. So I will take maybe a few students' questions. As a two sort of summary, I wanted to compare and contrast mesoderm and ectoderm. I think it's interesting to see how, these are very conserved processes. I think zooming into the subcellular mechanics, you can come up with a working model on how cells born alike become different. And in a nutshell, it's based on flow dynamics, which I argue is dependent on the ecadherine interactions. And, right. This is a bit pompous because I put principles, but I think we can argue. I think this is a sensible working model that recapitulates a lot of things found by different groups, ours included. And what I wanted to highlight is that flow patterns control the orientation of deformations, but you also need to control the stabilization. And we think that this information depends to some extent on this GPCR signaling, which I rapidly mentioned at the end, to be a common pathway for mesoderm and ectoderm. And I stop here. Many interesting questions, which you all have in mind. This is an old picture of the group. People have gone, others have come in. This is our Pierre-François, he's a great collaborator in Marseille. Okay, so any student question or not? Or coffee right now? Yes.
At the very ventral side of the embryo, where there are overlapping domains of, say, the pararoot genes such as Eve and odd and hairy expression, overlapping with the twist expression. So potentially there could be a conflict of direction regarding the deposition site of actin-myosin network, either to the apical side or to the junctional side, right?
Yeah, so I can make the conflict even ubiquitous. Remember the ectopic fog experiment? I put fog everywhere, so the conflict might be everywhere. Well, there's some dominance of fog. Cells now will not have junctional myosin II. They will accumulate myosin II apically, and indeed they will have mild apical constriction. So there is some kind of dominance based on ligands. We don't know why and how.
I see. So naturally, fog would dominate pararoot.
Somehow. It's a very sort of coarse description of a phenomenon that is, fog will override the activity of whatever makes planar polarity. And I would like to mention that, at this point, I sort of highlighted the point of planar polarity, but it's more adequate to say that there's two kinds of polarities. One is apical versus junctional. And that is what the GPCR signaling is doing. Of course, when it's junctional, when you lose the junctional, you also lose the planar polarity. But it's hard for us to tell at the moment whether this GPCR is also required to specify the planar cell polarity. Okay. It is definitely required for, but is it sufficient to convey this planar polarized information? That we don't know. And that's true of a lot of things in planar cell polarity. What is enough, sufficient to? Until we know where the ligands are and the way they work, it's going to be hard to. Okay?
So the germ is twisted in fog mutants, but do you know if intercalation itself is
Yes,
affected?
it's going on. Right. I think we go for coffee. Thanks.